All right, welcome back. In this video lecture, we're going to discuss muscle tissue physiology, which really means how does the actual contraction, right, of these muscle fibers occur and cause the muscle to contract. So let's start with defining what a motor unit is. So remember that we have somatic motor neurons, and these are the nervous system control over our skeletal muscles. So we have the upper motor neuron, right, that's in the motor cortex, and then we have the lower motor neuron. Most of those are going to be in the spinal cord, remember? And so if you think about the spinal cord, where was the cell body of that motor neuron located? So pause your video, see if you can remember that. Okay, so the cell body of the motor neurons are going to be located in the anterior great horn of the spinal cord. So anyway, that's just a review, but the somatic motor neuron is part of the motor unit, and the skeletal muscle fiber that it's innervating or that it's sending signals to is the other component of the motor unit. So motor units can be small. In other words, we have a few number of skeletal muscle fibers that are controlled, and this is going to be for muscles that have precise regulated contractions. So think about those extrinsic eye muscles, right, that help move your eyes. Those are going to be small motor units. Then we can have larger motor units, and the purpose of these, so you have many more skeletal fibers that are being controlled by the same motor neuron. And in this case, the whole point is for power, right? So think about your limbs, your legs, the muscles in your legs, the muscles in your arms. These are power generating contractions. Okay, next term is called a neuromuscular junction, and it is exactly what it sounds like. So neuro from the neuron, muscular for the muscle fiber, junction. So this is where that somatic motor neuron, right, is. So remember, there's not a physical connection. There's going to be a space there, but it's the place where the innervation is occurring between the neuron and the skeletal muscle fiber. Now, we'll look at a picture in just a minute, but the very end of the axon from the somatic motor neuron has what we can call a synaptic knob, or sometimes it's called a synaptic bulb. And that's where it sort of flattens out. And this is the end, right, where the action potential will finally reach the end of the axon. And what happens at that point is when that action potential travels to that region, it's going to open these voltage-gated calcium channels. So calcium then is going to come in down its concentration gradient. It's going to come into the axon, the synaptic bulb of the axon. And there are vesicles that are just located in this region, right, in the synaptic knob region. And these vesicles in the somatic motor neurons are going to contain a specific neurotransmitter. And this is something we need to know, and it's called acetylcholine, and it's abbreviated capital A, capital C, little h. So sometimes I'll use that shorthand notation. So what happens when the calcium comes into the synaptic knob, that's going to trigger these vesicles to fuse with the plasma membrane of the neuron, and basically exocytosis occurs, right? So it releases the acetylcholine neurotransmitter into this space, which is called the synaptic cleft. So the space between the neuron and the muscle fiber, this is the synaptic cleft. It's an actual physical space, okay? So also, I just want to make a note, later we'll talk about this more, but in this space exists an enzyme called acetylcholinesterase. This is an enzyme that the whole job of this enzyme is to break down or to degrade the acetylcholine neurotransmitter. And that's because we don't want, once it's released from the neuron, we don't want it hanging around forever, right? We want it released in response to an action potential. And then once that action potential stops, we want that neurotransmitter to no longer be in the cleft. So that's the purpose of the enzyme, to break it down in that cleft. Now, the part of the muscle fiber that is near this synaptic cleft is called the motor end plate. And in this area of the muscle fiber, there are many receptors that can bind the neurotransmitter acetylcholine. Now, these are called chemically gated or ligand gated ion channels. What that means is they function in response to the binding of a chemical or a ligand. In this case, this specific chemical they're responding to is the acetylcholine neurotransmitter. When that neurotransmitter binds to these receptors, that triggers the opening of these ion channels. Now, if you recall from when we talked about neurons and resting membrane potential, we know that there are more sodium ions outside of the cell than there are inside, and there are more potassium ions inside the cell than outside. So similar to a neuron, right, we have the sodium and potassium pump that's keeping more, so it's pumping sodium out against its concentration gradient. It's pumping potassium in against its concentration gradient. Then we have these leak channels, right, that are basically very important in maintaining that the inside of the cell is more negative than the outside. So as soon as you open ion channels, because sodium is more concentrated outside, it's going to rush in, right? It's going to move down its concentration gradient into the cell. Now, we know from when we talked about neurons, it's going to be the same idea. The resting membrane potential keeps the inside of the cell more negative. When you have positively charged ions coming in, right, it changes the charge inside the cell. All right, so this has got a lot going on in this image, but let's take it little by little. Let me turn my pen on here. So the first thing that I want to point out is this portion here, right, is the axon of the somatic motor neuron. And we notice that it is myelinated, right? So those signals are traveling quickly. They're jumping from node to node. The very end of that axon we see has the axon terminals that end in these flattened regions, right, that are called the end bulb or the synaptic bulb or the synaptic knob. Okay. Now, from the picture, it may look like those are making contact with the muscle fiber, but they're not. So if you look at a more magnified image here, we see that there's actually a physical space, right? That physical space between the neuron and the muscle fiber is called the synaptic cleft. So we have the end bulb, right, of the neuron, and then we have the motor end plate so this is from the muscle fiber, right? And the space in between is the synaptic cleft. Now, what we talk through is there are vesicles that are present at the very end that are containing our neurotransmitter acetylcholine. What triggers those vesicles to fuse with the plasma membrane and release the acetylcholine is as that action potential travels all the way to the end, right, and it reaches that synaptic end bulb, that triggers calcium channels to open, right? And calcium rushes in, and that's what causes the release of the neurotransmitter. Now, you can see once that happens, we have receptors on the motor end plate that bind acetylcholine. So here is an acetylcholine receptor, right, that's on the motor end plate. Once the acetylcholine binds, what that does is it opens those ion channels. So we know that sodium is more concentrated outside the cell. So as soon as those ion channels open, we're going to see an influx of sodium, which is a positively charged ion. So what does that do? Well, the inside of the cell was more negative, right? That's maintained, the resting membrane potential. But when we have positive ions that flow in, right, that's going to reverse that charge. So we'll talk about in just a minute what happens. So it's similar to a neuron. We have to reach, right, a certain threshold. Once that threshold is reached, then additional ion-gated channels will open. All right, so we mentioned briefly the resting membrane potential of a skeletal muscle fiber. So it's a little more negative than when we talked about the resting membrane potential of neurons. it's negative 90 volts. That means it's more negative on the inside by 90 millivolts, excuse me. So just like with a neuron, how is this resting membrane potential established? Well, it involves the sodium-potassium pump, right? Sodium-potassium pump is pumping 3 sodiums out for every 2 potassiums in. Remember, there are also negatively charged proteins that are present inside the cell that contribute to that negative charge. And then there are leak channels that are open that allow a small amount of movement, right, of ions. That's what establishes that negative 90. Now, we've already answered this, but which one of these ions is going to be more concentrated inside the cell at the resting membrane potential? So it's going to be potassium. Then which one is more concentrated outside the cell? That's going to be sodium. At resting membrane potential, the chemically gated channels, I'm going to put here ion channels, chemically gated acetylcholine ion channels are closed. Okay, that means before any acetylcholine has binds to those, right, the default is closed. Any voltage-gated sodium and potassium channels are also closed. That's the default, right? At resting membrane potential, they're closed. Okay, so up to this point, we've kind of looked at it. Big picture overview. Let's get down into the details, step by step, of how this happens. So the first thing that has to happen is we have to get the muscle fiber electrically excited. So as we mentioned, the motor neuron, right, is going to initiate this process because there's an action potential that travels down the axon. Now remember, this is review from our nervous system lesson, but remember, what is an action potential, right? It's when the voltage-gated sodium channels open, right, in a section. So let's say right here, right, in this axon, we have a section where the voltage-gated sodium channels open. Sodium ions rush in, right? That depolarizes the cell because it was about negative 70, right, is what a neuron is, millivolts at resting. And then when the sodium rushes in, that causes it to become positive, right? So that depolarizes the cell. And then after that, the voltage-gated potassium channels open and potassium will flow out. So when you have positive coming out, that repolarizes the membrane, right? And that triggers it to happen in the next section and the next section and so on and so forth, all the way to the end of the axon, right? So that's just review. That's how the action potential travels. At the end, though, when that action potential reaches the synaptic knob or the synaptic bulb, there are voltage-gated calcium channels at the end, right? When the calcium flows in, down its concentration gradient, that's what causes these synaptic vesicles to fuse with the plasma membrane. And when they do, exocytosis happens, meaning it releases the acetylcholine that was inside those vesicles into the synaptic cleft. Okay, so this is just a little more detailed look at what we've already talked about. Now we have receptors on the end plate, which is the part of the muscle fiber, right? That's a part of the synaptic cleft. There are receptors there for the acetylcholine, and acetylcholine then will bind to those receptors on the muscle cell. So that is how the motor neuron excites, right, the skeletal muscle by releasing acetylcholine and it binding to the receptors. Now, the second stage of a muscle contraction it's called excitation contraction coupling. So essentially, how does the excitation of the muscle cell actually cause contraction? And contraction is shortening, right, of the actual sarcomere. Okay, so how does this work? So once acetylcholine binds to those receptors, it's going to open ion channels. So these are chemically gated, right? The chemical that gates them is acetylcholine. Remember, the default is that they're closed. But when acetylcholine binds, it opens those channels, okay? Now, Here's the thing, just like with a neuron, right? There's a threshold that has to be met in order for an action potential to travel. It's the same with a muscle fiber. There has to be a change. So remember, with a muscle fiber, we're starting at negative 90 millivolts. We have to, or the muscle fiber has to reach a potential of negative 65 in order for an action potential to be initiated. If that negative 65 millivolt threshold is reached, okay, then we will see the propagation of an action potential along remember, the sarcolemma is the plasma membrane, right, of a muscle cell. That's what the sarcolemma is. The T tubules are these invaginations, right, that go internally into the muscle fiber so that they actually can make contact with the sarcoplasmic reticulum. So the action potential will travel along the plasma membrane or the sarcolemma and the T tubules. In the same way that an action potential travels in a neuron, right, you get sodium ions flow in first, that depolarizes that area. And then after that, we get repolarization with the calcium ions flowing into the cell. And the same process, right? It's traveling one segment that goes through this depolarization and repolarization, initiates the next segment, and it just goes on in a domino fashion. Now, how does this lead to the actual contraction? Okay. The way it leads to the contraction is once that action potential reaches what's called a triad. So that's where the T tubules make contact with a portion of the sarcoplasmic reticulum. Okay. Remember, that's the fancy endoplasmic reticulum. That's our fancy word for a little specialized endoplasmic reticulum in a muscle fiber. Once it reaches what's called the terminal cisternae, so that's a portion of the sarcoplasmic reticulum that is next to the T tubules. So we have a T tubule with a terminal cisternae on either side. So it's like 3. That's why it's called a triad. When that happens, it triggers calcium ions to be released by the sarcoplasmic reticulum. Okay, so in this image, okay, this is a T tubule that we see coming down. Okay, this would be it's going to take me just a minute. The terminal cisternae of sarcoplasmic reticulum. Okay, terminal cisternae of this. Now, what I want you to see is the muscle fiber always pumps calcium so that we have a store of calcium inside the sarcoplasmic reticulum. When that action potential travels through the T tubule, it triggers calcium ion channels to open in the sarcoplasmic reticulum, and then calcium is just going to travel down its concentration gradient, okay, which means what's outside of the sarcoplasmic reticulum, okay? This is just the sarcoplasm, right? Or the cytoplasm of the muscle fiber, okay? So calcium then, so far, what we've gotten to is action potential travels through the T tubules. That causes the calcium ion channels to open. They're voltage gated, right? It causes them to open in the sarcoplasmic reticulum, and calcium ions, I'm just going to put here, calcium ions are released into the sarcoplasm. Okay. Now we're going to look at how is the release of calcium going to cause the actual contraction of the sarcomere. All right, so the next step, which would actually explain how the sarcomere contracts, is called cross-bridge cycling, and it has to do with the myosin and actin filaments. However, there's a regulatory piece that prevents that from happening. And this regulatory piece has to do with 2 proteins that are part of the thin filament, right? We talked about the thin filament. The primary protein is actin, and that's true. But what we see in this image is, along with actin, there are two regulatory proteins. One is called troponin, and it's right here, and the other one is called tropomyosin. So these are present along the actin or along the thin filament. They prevent the thick filament or the myosin from binding to actin because of their position. So essentially, there is a binding site for myosin that is being covered up by the troponin and tropomyosin proteins. When calcium is released from the sarcoplasmic reticulum, calcium will bind to the regulatory protein troponin. Essentially, what calcium binding does is it causes troponin to change shape, and it moves that entire troponin-tropomyosin complex so that it reveals or opens the binding site for the myosin heads. Okay? So we move from this direction to this right here. Myosin cannot bind because the regulatory proteins are covering the binding site. When calcium binds, it shifts those regulatory proteins, and now we see that myosin can bind to actin because that binding site is now free or open. So that is the reason that calcium is so important. Without calcium being released from the sarcoplasmic reticulum, we can't have the shortening of the sarcomere. Okay, so now calcium removed the regulatory proteins. We'll look at how does this cross-bridge cycling happen. So it's a multi-step process, okay? And it's going to involve both the thick and the thin filament. So when you think about the sarcomere, remember we had the sarcomere that was, we had these Z discs, right, on either end. And in between each Z disc, we considered that a sarcomere. And we had these regions where it was only the myosin thick filaments, and where there was overlap between the thick and the thin, and when there was only the thin filament. So we're looking at a very magnified view, right? The purple is the myosin or the thick filament, and the green is the thin filament or the actin. Now in cross-bridge formation, the myosin that has this head structure, okay, the thick filament, is going to bind to actin. Now remember, this can't happen without the presence of calcium to remove those regulatory proteins. Now, so far, nothing has shortened, right? We just have the binding of the myosin head to the actin. In the next step, it's called the power stroke. In this step, the myosin head actually shifts toward the center of the sarcomere. And because it's attached to actin, when it shifts, it is pulling the actin filament toward the center of the sarcomere. When it does that, notice initially when it first bound to actin, it has ADP or adenosine diphosphate and a phosphate molecule attached. ADP and the inorganic phosphate are released during this power stroke portion when it actually shifts and moves the actin. The next step is called the cross-bridge detachment, and it is exactly what it sounds like. That's when the myosin head releases actin. Now, it cannot do that unless there is ATP present. So remember, if you remember back when we talked about the cell and energy, ATP is the high energy molecule, right, that our mitochondria are producing all the time. So what happens is ATP binds to myosin, and then myosin can release the actin filament. The final step is to basically reset or to, quote, cock the myosin head back into place. And in order to do that, then the ATP is hydrolyzed or broken down into adenosine diphosphate and an inorganic phosphate. That's what returns it back to its initial position. Okay, and then as long as the action potential is being received and calcium is still being released and there's ATP present, we will have this cycle, right, happen all over again, over and over and over. Okay, so big picture, what's happening, the actual filaments are not changing their size, right? myosin doesn't get shorter, actin doesn't get shorter, but the sarcomere does get shorter. And that's because, remember, what's happening is, if you can imagine this top one, this is when the muscle or the sarcomere is relaxed. Every one of these myosin heads is grabbing on, right, to the actin filament on this side. And as those heads are shifting, right, it is moving the actin filament toward the center. On this side, the myosin heads are binding, right, and they're shifting the actin toward the center in this direction. So the result is what we see down here. The filaments themselves are not shorter, but they slide past one another, right? The actin slides toward the center because the myosin heads are moving it toward the center. And so the Z bands originally were here, right? Or the Z discs, and now they're here. So the overall sarcomere shortens because those filaments slide past one another. That's where they get the term sliding filament model, because those filaments are moving past one another. Now remember, the banding patterns are what we see that we call striations. So what's happening to these different bands? Let me erase a little bit of my marks on here so we can see the bands better. Okay, so the midline, right, is still the midline. That doesn't change, obviously. The A band, okay, which is the darker one that contains the myosin is not changing shape or size. So the A band stays the same. The I band is the lighter part that only contains the actin, right? No myosin. So as the actin is pulled toward the center, that I band is getting smaller and smaller and smaller, right? Because we have those filaments overlapping. Now, the H zone gets smaller. The H zone was the area in the center that only had the myosin and no actin. As the actin slides toward the center, right, then obviously that H band is also going to get smaller and smaller. But again, the actual filaments don't shorten, right? The sarcomere does, but not the actual filaments. Okay, so this exercise is also in your study guide. So I would highly recommend stop the video, put these in order yourself first, and then turn it on and check yourself. Okay? So first thing that happens, in at least the list that I have here, right? We have to have the neuron, so the somatic motor neuron, releasing our acetylcholine. That's number one. Now, number two, the chemically gated sodium channels have to open. Okay? And then three, the membrane of a muscle fiber depolarizes. So I just want to so this part, right? Acetylcholine is released into the synaptic cleft. The part that maybe it skips a step is bound, right? The end plate of the motor neuron is going to bind that acetylcholine. That's going to open, right? When it says chemically gated, it's a specific chemical, right? The chemical that is triggering that is acetylcholine. That opens those sodium channels, okay? Now, the membrane of a muscle fiber is going to depolarize, right? As long as the threshold is met, it's going to depolarize. Number four, when that depolarization reaches the triad, right, where it travels down the T-tubule and it reaches the terminal cisternae of the sarcoplasmic reticulum, that's going to trigger these calcium channels to open, okay? These are voltage-gated calcium channels. When the voltage-gated calcium channels open, then we get those calcium ions binding to the regulatory protein troponin. Then we see that whole troponin tropomyosin moves, right? Shifts, it changes shapes and shifts and opens up the binding site for myosin on actin. And finally, the myosin is going to attach to actin, and the whole cross-bridge formation shifts and causes the shortening of the sarcomere. Now we know that we don't contract our muscle and just it stays contracted, right? So muscles are contracting and then they have to be able to relax. So once the motor neuron stops sending the action potential, right, then the release of acetylcholine doesn't happen anymore. However, there will be some acetylcholine that's still present in the synaptic cleft. Remember earlier we mentioned this specific enzyme called acetylcholinesterase, and the whole purpose of this enzyme is to break down or degrade acetylcholine in the synaptic cleft. So once the release stops, right, this is going to be broken down by this enzyme. When there's no acetylcholine, those chemically gated ion channels don't open at the end plate, so threshold is not reached. Action potentials in the muscle cells stop. If there's no action potential reaching the sarcoplasmic reticulum, there's no release of calcium ions into the sarcoplasm. And there are pumps that are constantly moving that calcium back to the sarcoplasmic reticulum. If there's no calcium to bind troponin, then those two regulatory proteins are going to cover over that binding site for the myosin head on the actin, and we will not have cross-bridge formation and we will not have contraction. Now, lastly, let's talk about rigor mortis. So we know, because we looked at how cross-bridge formation happens, we know that ATP has to be present or we can't have muscle contraction. But think back to which step of that cross-bridge cycle actually uses ATP. Well, it's actually the point that allows myosin to release actin. So after death, right, there's a certain amount of ATP in the cell. Once that ATP is completely utilized, obviously there's no more ATP being generated, and therefore there's not enough ATP eventually for the myosin to release from actin. So it's stuck, right, in that bound state. The other thing is, remember I mentioned that calcium is constantly being pumped back into the sarcoplasmic reticulum from the sarcoplasm. That takes energy. When anything is being pumped, that means it's requiring energy of the cell. So to pump calcium out of the sarcoplasm takes energy and ATP. So rigor mortis happens once the ATP is all hydrolyzed and those muscles then are stuck in the contracted form. And that's going to last maybe up to 24 hours until the actual muscle fiber tissue starts to be broken down or hydrolyzed. So I'm sure you know, if you watch true crime or if you've read about it, that's used as when a body is found, right, based on the temperature of the body and what part of rigor mortis, to try to identify how long the body has been dead.